All right, so where we left off on the last video was talking about the cell cycle. Okay, now the cell cycle is basically the life cycle of a cell. We last one we talked about the structure of DNA, how it's the different characteristics of it. And we talked about why it's important. The instructions to carry out the cell's functions are housed within the DNA. Okay, and so when a cell divides, it has to be able to pass down those instructions to the new cells. All right, and so before it can divide, and we'll talk about cell division next week, but before the cell can divide, it has to replicate the DNA. So if you look at the life cycle, most of the life cycle is in this larger interphase. And the interphase can be divided into G1, S, and G2. Okay, well, G1 is just normal cell living and maintenance. Okay, and as you see, it spends most of the time in G1 of interphase. Okay, but as it gets ready to divide, it goes into S phase. All right, and the reason why it's called S phase is because you have DNA synthesis. Well, in the S phase is where you have DNA replication. All right, and then after it replicates the DNA, it will go to G2, where it makes sure it goes through a number of checkpoints to be like, all right, am I ready to divide? And if it meets those criteria, then it will proceed to mitosis. All right, but again, before it can go into mitosis, before it can meet those checkpoints, it has to replicate the DNA in the S phase. Okay, so let me go to presentation view. All right, so how does the cell copy its DNA? All right, so in preparation for division, the cell copies its chromosomes so that it contains two sets. All right, because the new cells that are produced during mitosis, it has to have a complete set of DNA or a complete set of chromosomes. All right, and so that the parent cell, which then creates the two new cells, has to give each of the new cells a complete set. All right, in order to do that, it has to go through DNA replication during the S phase. All right, so this is kind of the nuts and bolts of DNA replication. All right, DNA, as we said in the last part, is double-stranded. So the double-stranded DNA molecule separates, and both strands of the DNA molecule acts as templates for the new strand. So if you look at the new DNA molecules that are produced during DNA replication, one strand is old and one strand is new, okay, in both DNA molecules. Okay, and so that's why it's considered semi-conservative replication, because when you look at the new DNA molecules after DNA replication is done, one strand is the old strand that came from the parent DNA molecule, all right, that acted as a template to produce the new daughter strand. So semi-conservative replication. Now, DNA replication happens at multiple places within a DNA molecule. So you'll have what's called a replication fork. Here you also have a replication fork further down on the DNA molecule. Now remember, the DNA molecule has orientation, so the 5' end is always bound to the 3' end on the other strand. Okay, so that's what those numbers are for. But those replication forks will continue to grow, where you have the new DNA strands being produced from the old DNA strands acting as templates. And eventually, all right, these bubbles enlarge in both directions and they join up. And so when they join up, it means that two new strands, these dashed lines, have been created. And so now you have two new DNA molecules. Each DNA molecule consists of an old parent strand, the template strand, and the new daughter strand. Remember, we said that the DNA molecules are complement, or the strands are complementary to one another. All right, and so that's why these old strands act as templates, because what will happen is, as the new strands are created, okay, the sequence, the base pair sequence on the daughter strands are complementary to the parent strand. All right, and as we said in the last part, A always binds to T, and G always binds to C. So if you know the sequence of one strand of DNA, you know what should be the sequence of the other strand, because they're complementary to one another. All right, so now let's talk about DNA replication, okay? Because DNA replication looks differently depending on what template strand you're looking at. All right, so one template strand is called the leading strand, and one DNA strand is called the lagging strand. So in this figure, you have this one, okay, the leading strand, and the leading strand, it runs 3' to 5', okay? All right, so the leading parental strand, the orientation of it runs 3' to 5'. Okay, the other parent strand that runs 5' to 3' is called the lagging strand, and we'll talk about that in a minute, okay, why that is. Because DNA replication looks different depending on if you're talking about the leading strand or the lagging parental strand. Okay, so we're first going to talk about on the leading strand. Okay, before we even get into leading and lagging, some enzymes come in first, okay? So here you have topoisomerase and helicase, and we know they're enzymes, right? Because they end in that suffix ASE. Okay, so topoisomerase unwinds the DNA strand. All right, remember that DNA strand is DNA is a double helix. Okay, so you got to unwind it to make it more looking like a ladder. Then helicase will come in and breaks the hydrogen bonds. Where are those hydrogen bonds? They're between the nitrogenous bases. Okay, so first that happens at each replication fork or bubble. All right, now let's talk about the leading strand. Now you have this enzyme, DNA polymerase, that is the main enzyme to come in and add bases to the growing DNA strand. But it can't just start building a DNA strand out of scratch. It's gotta have a head start. It's gotta have something to bind to first and add the base pairs to. Okay. Well, when you look at the first part of a new daughter strand, okay, you have a little piece of RNA. So RNA primers will come and sit on the 3' end of the parent DNA strand. And then that DNA polymerase, that enzyme, can then come and sit on the RNA, okay, and then start adding complementary base pairs to the growing daughter strand. And DNA polymerase can only work in one direction. It can only work 5' to 3' in reference to the daughter strand. Okay, so here, if we go back here, so if the parent strand is 3', then the daughter strand is going to be 5', right? All right, so here you have RNA right here, and then DNA polymerase will come in, sit where this RNA is, and then start adding in this direction complementary base pairs. And it can work, all right, continuously, because it can work 5' to 3'. Okay, DNA polymerase will sense, okay, what's this base pair? Oh, that's adenine. All right, that means a thymine has to go here. Oh, is this a thymine? Then adenine has to go here. Oh, this is a cytosine, then a guanine has to go here. So it reads what the base pair of the parental strand is at each location, and then adds the complementary base pair to the daughter strand. All right, and again, it can only work 5' to 3'. All right, how does it get energy to add those, to make those bonds, right? Because whenever we're making chemical bonds, we talked about at the beginning, you have to have an input of energy. Okay, well, it gets that energy to add the base pairs, the nucleotides to the growing daughter strand by breaking off a phosphate group from the nucleotides that they're adding. Okay, so again, this is just another figure, okay, showing you DNA replication. So if we're thinking DNA replication is going this direction, okay, all right, this would be the leading parent strand, this dark blue, okay. So that means what's the orientation of this parent strand is 3' to 5'. So DNA polymerase is sitting on it and adding complementary base pairs on the daughter strand running 5' to 3'. Okay, well, but it also has to do it on the other parental strand lagging, all right? Well, here we have a problem, right? Because DNA polymerase can only work 5' to 3', okay? And so if the overall direction of replication is here, okay, we're having a problem because DNA polymerase can work on this parent strand only in this direction, okay? So if overall direction of replication is going from right to left on this lagging strand, okay, DNA polymerase can only work from left to right. So how does that happen? How does DNA replication occur on the lagging strand? Well, it occurs discontinuously. So on the leading strand, the daughter strand is made continuously, but on the lagging strand, it's made in fragments. It's made discontinuously. All right, so what happens is instead of having just one primer at the beginning of the daughter strand, there are multiple RNA primers that are attached to the lagging parental strand. And what that allows the DNA polymerase is it sits on an RNA primer and then works in the direction it does. And once it gets to where the daughter strand is now, it will drop off and then go to a new primer and work backwards. Okay, once it hits the daughter strand where it had just worked, it'll drop off and go to another spot. Okay, so it works discontinuously and makes fragments. Okay, so the daughter DNA strand of lagging DNA parental strand is made in fragments. The RNA primers are made at different spots along the lagging parental DNA strand, and so that DNA polymerase sits down and adds complementary nucleotides on the daughter strand until it hits the previous fragment. Okay, all right, and then it will drop off and then move forward to another primer and then work backwards until it hits the previous fragment. It will drop off and then go forward, sit on a primer, and work backwards. All right, and those fragments actually have a specific name. They're called Okazaki fragments. All right, because there was a married couple named Okazaki who actually discovered this. Okay, discovered how DNA replication was done on the lagging strand. All right, well, DNA polymerase doesn't bind those fragments together into one new daughter strand. There's another enzyme called ligase. The ligase will come in and bind the Okazaki fragments together to make the full daughter strand on that is complementary to the lagging parental strand. Okay, so it's hard to do a good job explaining this when I'm not standing in front of you and drawing on the board. It may be really hard for you to understand it just by looking at a figure, and so that's why you need to spend some time thinking about this and looking at it. Look at some visualizations. Okay, you'll see that on the Edpuzzle. You can look for other visualizations on the internet because it's a little tricky thinking how DNA replication happens on the lagging strand if it's not sort of almost done in motion for you. All right, so that's the basics of DNA replication on both the leading and lagging strand. All right, so every DNA molecule that's in a cell has to undergo that process so that they can be replicated. And again, the whole point of DNA replication is so that a cell, when it divides, it can give a complete set of DNA to each of the two new daughter cells. All right, well, as you can think about, there are, hundreds, thousands, millions sometimes of nucleotides within a DNA molecule. All right, and so the more nucleotides that are needed to be added to a DNA molecule, the more chances of mistakes happening. Okay, DNA polymerase is not perfect. Excuse me, it's not perfect. Sometimes it does make mistakes. All right, now there's a proofreading mechanism that happens that it has to be like, okay, I put a T there when I should have put A, or I put a C there when I should have put a T. All right, so it can sometimes go back and fix its mistake, but sometimes it doesn't. Okay, and sometimes that mistake is permanent. All right, and so what do scientists call that? A mutation. A mutation is a permanent change in the DNA sequence. All right, the replication errors may occur after a cell's DNA gets broken or damaged, and because DNA polymerases don't copy damaged DNA very well. All right, so we'll talk about some things that can damage the DNA inside a cell. But when proofreading and repair mechanisms fail, that error becomes permanent and becomes a mutation. All right, so here you have through replication, somehow an A gets added here, all right, when it should have been a T. Okay, this A should have been a T, right, because it's base paired with this A right here. All right, but if this mistake is not corrected, once this DNA molecule gets replicated, here that's a permanent mistake. Okay, where this A that should have been T, all right, is now permanently A, and this T that should be A is now changed to T. You have a permanent change in this DNA molecule, which will then be replicated for future generations. All right, so what can cause DNA damage? You have radiation, all right, ionizing radiation from X-rays, most UV light, and gamma rays may cause DNA damage. And when we talked about the electromagnetic spectrum and photosynthesis, all right, did X-rays, UV light, and gamma rays, did they have short wavelength or long wavelength? All right, these were shorter wavelength energy. All right, and as we said, the shorter the wavelength, the more power it has. All right, so these are some very powerful radiation waves, and that can cause DNA. So that's why when you go to the dentist and you get X-rays done on your teeth, they'll put a lead apron over you because they don't want to expose any more cells than they have to those rays, right, that could cause damage. All right, UV light from the sun. That's why you need to wear sunblock. All right, to help prevent damage to your skin cells. All right, so these ionizing radiation waves breaks DNA, and they cause covalent bonds to form between bases on opposite strands. They can fatally alter nucleotide bases, all right, and then they can cause adjacent nucleotide dimers to form. All right, and so mutations can form in any type of cell, and those mutations that occur in egg and sperm cells will be passed on to the offspring. All right, mutations that alter the DNA instructions may have harmful or lethal outcome, and that's most cancers begin with mutation. All right, so if we go back to this idea of UV radiation from the sun, all right, those can damage to skin cells' DNA, which can lead to the formation of cancerous cells, skin cancer. All right, but not all mutations are dangerous. You usually think of mutations of being negative, but not always, because mutations also give rise to variation in traits, which then give rise to biodiversity, give rise to evolution. Okay. All right. So, again, we talked about how certain agents of mutations can cause. So here you have chromosomes that have been exposed to radiation, and you see breaks in those chromosomes, and these damaged chromosomes won't be replicated very well by DNA polymerase, and so they'll be more prone to having mutations in them. All right, and again, when we think of mutations, we usually think of this like a frog with 3 legs, and that can happen due to different chemicals that can lead to mutations. All right, so need to know, what is the structure of a nucleotide in DNA? Okay, what are the basic structures of a nucleotide, and what's the basic structure of DNA? What does complementary strands mean? And if I give you a DNA strand, okay, can you provide the complementary strand of DNA? You should. All right, how is DNA organized in the cell? When we talk about chromosomes and chromatin. All right, now you also need to know the basics of DNA replication. What are those enzymes involved? All right, what's the difference between what happens on the leading strand and what happens on the lagging strand? Why is it considered semi-conservative? And then what are mutations and what can cause them?